All right, welcome to the review for Bio 144. This is for the fourth exam, which is going to be our final. As soon as I start getting questions, I'll start talking about the slides, and we can review anything from either the most recent chapters with the nervous system, or we can go back over old material from the cumulative study guide as well. So as soon as I start getting questions, I'll start talking about some science stuff. All right, looks like I just had some pretty great questions come in about changes in membrane permeability and generation of action potentials. So let's go ahead and start there. I'm going to pull my slides up and we'll talk about action potentials first. Okay, so give me 1 second, just going to check and make sure that the sound is coming through all right. Oh, sounds beautiful. All right. So we're going to start kind of in the middle of chapter 12, dealing with action potentials here. So here's the first bit of information that we have about action potentials, is that we understand that we're dealing with the electrical potential of a cell. So normally our resting membrane potential, which is meaning that inside the cell we're going to be at a negative 70 millivolts, as we can see over here on this side of our graph. And let me just double check that I can actually, that all of you can see my mouse. How interesting. Let me go ahead and just change one thing so that you can see the mouse a little better. Beautiful, there it is. All right, I'll make this a little bigger. Okay, so when we're talking about action potentials, I'm going to really quickly review action potentials and what goes on with action potentials, and we'll talk about what's happening at the membrane for each phase of the action potential. So our action potential is always going to start with some stimulus that's going to push us up to negative 55 millivolts. We have to meet that threshold before an action potential starts. If we don't meet the negative 55 millivolt threshold, nothing's going to happen. So we have to meet this threshold, and once we do, we're immediately going to go into an action potential. And as you remember, action potentials are what we call all or nothing. So they either happen or they don't. So for an action potential, once we reach that threshold, we're immediately going to go into this very fast depolarizing phase, which is going to push us up to plus 30 millivolts. And then right as we hit that peak, we're going to go into the repolarizing phase. We're going to actually overshoot our normal resting membrane potential, and we're going to drop down into this after hyperpolarizing phase. So we're always going to have this kind of graded potential up to our threshold. Then our action potential is this spike, drop, and then the repolarizing after we do our after hyperpolarizing phase. So what we can see over here to the side is what exactly is opening when. So here in this first phase, in this kind of purple shaded area over here, this is showing us that our voltage-gated sodium channels are in the resting state, and our voltage-gated potassium channels are also closed. So our voltage-gated sodium channels look like this. So our sodium is going to be able to come through when they're open, but we have two different ways that we can open and close the gate. So our activation gate here, normally when we're doing an action potential, this is what's going to open. And then all of the sodium is going to come rushing in, and then that's going to happen over a couple milliseconds. Once it's activated, we're going to have this all flood in, and these are voltage gated. So once we hit negative 55 millivolts, that's what it's actually triggering, is it's triggering this. So there's a rapid opening here that's triggered at the threshold, and then we have a slow closing afterward that's also triggered at the threshold. So this is going to happen at the same time, but it's going to be much more slow, which is going to give enough time for the sodium to come rushing into the cell. This here, this slow closing of this inactivation gate is what is actually going to cause the refractory period at the end of the action potential. So this is one thing that we're going to do at the start of our action potentials. We're going to open these gates. We're also going to open, it doesn't look like I actually have a picture of our potassium gates. That's fine. So with our action potential, we're going to open both sets of gates. And it takes the potassium gates a little bit longer to open. So the sodium gates open first. All the sodium is going to rush into the cell. And this here is going to happen after our blue phase, which is, again, where we depolarize to the threshold. And depolarizing means we get closer to zero, or we get more positive. So this whole first part is a depolarization, hence we call it our depolarizing phase. And then our voltage-gated sodium channels are going to be open, so sodium is going to rush into the cell. So we have this massive amount of positive ions all rushing into the cell, which is going to make the inside of the cell a lot more positive. If you go ahead and give me 1 second here, I just want to make sure that everything is working as it should. Sounds like it's all working. I just want to double check given how many issues we've had in the past. So here's our action potential again. Back to here, we've gotten halfway through it. So far what we've been dealing with is our voltage-gated sodium channels are open here, and so they're open. They've been open, and then now they're going to start closing here at the peak of our action potential. Our voltage-gated potassium channels are going to take a while to open. They're going to get triggered here, but they're not going to actually open until the peak of this action potential curve. So up here is when our potassium channels finally actually open. So once they open, all of the potassium is going to rush out of the cell. So we had all these positive ions rush into the cell. Now we have all these positive ions rushing out of the cell. And the reason that we can't have more sodium coming in is because our sodium channels are inactivating over here. Okay, so this is where our sodium channels are inactivating, is going to be during the repolarizing phase. So our repolarizing phase is when we're going to shoot down here, we're going to hit the threshold, and then we're going to enter this big orange phase. This is our after hyperpolarizing phase, and it's also going to be our relative refractory period, where our voltage-gated potassium channels are still open, so they're still going to let the potassium out and out and out, which we're actually going to, as we said, overshoot that -70, and our sodium channels are still in the resting state, so they haven't been put back to an activatable state yet. So they're not quite ready. Those voltage-gated sodium channels haven't gone back from this form to this form quite yet. So that's when we're in a refractory period right here. Once we then figure out, once the cell figures out to actually close all of its channels, we're going to close this channel right here. This is going to be our potassium channels, because our sodium channels have already been closing this whole time. Our potassium channels are going to close, and then the sodium-potassium pump is going to return us to our resting membrane potential. We're going to push all of that sodium outside the cell. We're going to put the potassium back in the cell, but remembering that there is something that's really key about this resting membrane potential, and it's that the permeability is different for sodium and potassium. So once we push that sodium outside, it's going to stay there. But potassium, we're pretty leaky for potassium, so it's pretty easy for potassium to actually get into the cell. So it's always kind of in a state of flux, and that's why it's easier for us. Oh, I completely lost my train of thought. Where was I going with this? This is what allows us to return back to our resting membrane potential here. When we go a little bit more negative, we're going to get the sodium basically where they belong. That's going to reset our resting membrane potential, where we have all these positive charges on the outside, all these negative charges on the inside, and the potassium is going to be in a bit of a state of flux. Generally, we're going to have more inside the cell than outside, but that limit isn't quite as hard of a limit as it is with the sodium ions. So let's see, changes in membrane permeability. Let me just go through these slides and make sure that I figured that out. So as far as applying everything that we just talked about to, let me flip back here. Applying everything that we just talked about to membrane permeability, what we're going to see is we can go through this graph kind of with a couple different ideas in place. So we can go through this thinking about what is going to be the membrane permeability to sodium, what is membrane permeability to potassium, and kind of go through it like that. So here, our membrane is going to be very much not permeable to sodium. It is going to be pretty much completely impermeable to sodium. It's not going to be able to get through. There's no leakage channels for sodium here. Then all of the sodium channels are going to open, and in this depolarizing phase, the membrane is going to be incredibly permeable. We are going to be very, very permeable to sodium during this phase. Then we're going to be incredibly permeable to potassium during the repolarizing phase until we get to the bottom of our after hyperpolarizing phase. Oh, that is a mouthful. And then once we start going back to resting membrane potential, we are no longer going to be as permeable to potassium. And as I mentioned, there are a lot of leakage channels, so the membrane is pretty permeable to potassium in comparison to sodium. But here it's much more permeable to potassium than normal. So we kind of have this wave of really permeable to sodium, and then we stop, and then we're really permeable to potassium, and then we stop and we reset. And once we get back to this resting membrane potential, we get back after we finish this after hyperpolarizing phase, we go fully through the refractory period. Now if there is another graded potential that takes us up to the threshold, now we could fire another action potential. However, if we give the cell another stimulus anywhere in this area, the cell cannot respond to another stimulus. It has already fired an action potential. It cannot fire another one until it gets over into the resting membrane potential again, and the sodium ions are fully outside, the potassium ions are fully inside, and we go back into this form for our voltage-gated sodium channels. Once they all switch from this to this, then we can fire another action potential, which is a good thing because action potentials are really important, as we know, and This is only lasting a couple milliseconds. So this is 2 to 3 milliseconds. So it's pretty easy for us to fire one active potential and then another active potential because they don't take that long to actually fire. Oh, that's the wrong window. Okay. We've got a question here. Okay, describe up and down regulation in regards to hormones. Ooh, I'm going to have to pull open some new slides for that. So we'll do up and down regulation, and then I'll jump back into the questions that I got emailed. And we'll just bounce around as we need to. Okay, so let's go into our chapter 18 slides. And we're going to talk about the role of hormone receptors. So this is from the review stuff. This is from the cumulative material. This is specifically from chapter 18. So when we're thinking about hormone receptors, we have to remember that hormones are only going to have an effect on target cells. So if there is a cell that has a receptor that can basically hear the message that this hormone, that this endocrine signal is sending, then it can respond. If it doesn't have a receptor, it cannot respond. So when we're dealing with downregulation or upregulation, what we're going to be dealing with is the response of the receptors on the target cell. So some information that we have here in front of us, we can see that we're going to have a range. We're going to have anywhere from 2,000 to 100,000 receptors per target cell, either on the cell surface or in the cytosol, depending on the type of hormone, if it's hydrophobic or hydrophilic. So what we see is there are a couple different situations that can arise. If we're making too much hormone, so if we have an excess of hormone, we basically have too much hormone being produced here, and we're overstimulating the target cells. So if we're overstimulating the target cells, the target cells are going to respond by reducing the number of receptors that they have, so that they're still getting the correct amount of a message. But what this results in is that if the hormone then drops down from excessive levels, it's no longer as sensitive. So if we have excess hormone being produced, that's going to cause a decrease in the number of receptors on the cell. And this is something that we actually see in diabetes type 2, where we're overproducing the hormones that are responsible for blood sugar levels. And this is going to overall make the target cell less sensitive to the hormone. So this is that we have a really high level of hormone. It's going to reduce the number of receptors that the cell has because it's being overstimulated. And then if the hormone levels drop back down to regular levels, it's not going to seem like, quote, regular levels to the cell, because that cell is no longer as sensitive as it should be or as it was before, because it has lost those receptors. The other situation, which is kind of the inverse of it, is upregulation. And upregulation is the deficiency of a hormone. So when we talked about hormones, we compared them to messages and to information. So for upregulation, if upregulation is, if we're dealing with that hormone message metaphor, then upregulation is going to be a situation where somebody is whispering, right? So when somebody is whispering and you can't really hear them, you're going to lean in, you're going to try to be more sensitive to what they're saying because you just can't hear them. And that's what happens with upregulation, where you don't have as much hormone as you did before or as you should, so these cells are going to produce an increase in the number of receptors so that they get more sensitive to the hormone, so that if the hormone is present, even in very low levels, they're very sensitive to it. If the hormone levels then go back to normal, your receptors are now going to be, your cells are now going to be hypersensitive to the hormone. So upregulation is like if somebody is whispering and you lean in to hear them, and then if you lean in and suddenly they start talking at normal level, you're going to be like, oh, that's so loud, it's way too loud, which is what happens to the cells if the hormone levels go back to normal. Downregulation is like if someone is yelling at you, right? You're going to step away, maybe put your fingers in your ears so you can actually hear them at a regular level, but then if you've walked away or you're in the next room and they start talking at a normal level, you're not going to be able to hear them. So downregulation is if you make too much hormones, or you make too many hormones, too high of a level of hormone, and you decrease the number of receptors, which decreases the sensitivity. Upregulation is if you don't produce enough hormone, you're going to get an increase in receptors, and that's going to make you more sensitive to the hormone. Okay, hopefully that helped. And let's go. Okay, I don't see any other questions in the live chat, so I'm going to go ahead and go back to the questions I was emailed. One of them is the anatomy of a chemical synapse. And what exactly does that mean? Let's figure that out. Where do I want to be right now? That's going to be the later chapters. That's going to be, is it the later chapters? No, it's the earlier chapters. That's still chapter 12. So much information in chapter 12 that we can then apply to the later chapters. I almost didn't realize exactly how much time we spent there. We sat on there for a long time. We can start here. Okay, so I think one of the things that I pointed out or asked you to understand, or might be in one of your study guides, is the anatomy of a chemical synapse. So what is a chemical synapse? What does it look like? How does that affect what it does? So for a chemical synapse, the important thing to know is that if it's going to be a chemical message, there has to be a physical barrier that we're crossing. So there's two different ways that we can get information from one neuron to another. One way to get that information from one neuron to another is electrical information. And we talked about this with cardiac cells and with some neuronal cells where we have gap junctions. And the ionic current can just spread directly from one cell to another. The other type of information that we mostly focused on is chemical synapse, which is the way that we get information from a presynaptic to a postsynaptic neuron. So here's what that synapse looks like through time. So we'll do basic anatomy and then we'll look at it through time. So here's one of our axons. This one is the one that's currently conducting an action potential down its axon. And when the action potential gets down its axon, that's going to trigger this voltage-gated calcium channel over here. Sorry, I'll stick to the anatomy first and then we'll do the physiology. So this is our axon over here of our presynaptic neuron, and then we have this big kind of foot. This is our synaptic end bulb, also called our synaptic end terminal or the axon terminal. This nice big bulge right here. And then this protein is a voltage-gated calcium channel. The space between the presynaptic neuron and the postsynaptic neuron is what we call the synaptic cleft. And then we're also going to have neurotransmitter receptors that are attached to these ligand-gated ion channels. So what's actually going to happen, as I was already talking about, is that we're going to have this action potential run down to the end of the axon, and as the action potential gets to the end of the axon, that's going to trigger this voltage-gated calcium channel. As we've already seen with some other voltage-gated channels, if you change the electrical state of the inside of the cell to some threshold, that will then trigger the voltage-gated channels. So in this case, they're voltage-gated channels and they're voltage-gated calcium channels. So when this action potential gets down here and we have this really rapid depolarization, repolarization, that depolarization is going to cause this calcium channel to open. We're going to get calcium to flow inward. That's going to trigger the release of this neurotransmitter, whatever that neurotransmitter is. We went over many different kinds of neurotransmitters and generally what they do and when we see them. And so these neurotransmitters are going to be stored in what we call synaptic vesicles. So this is where we're going to have vesicular transport. So they're going to be wrapped up in some membrane. The calcium is going to come in, and when that happens, we're going to have this little ball of membrane fuse with the outer membrane that's going to release the neurotransmitter into the synaptic cleft. When that neurotransmitter gets past the synaptic cleft, this is going to be one of three locations. This postsynaptic neuron, this can either be the axon hillock of another axon, this can be the cell body, or this could be the dendrites. So this can be either an axo-axonic, an axodendritic, or a I think it's an axosomatic, yes, axosomatic type of connection, where axodendritic is going from an axon to a dendrite, axosomatic is from an axon to a cell body, and axo-axonic is going from an axon to an axon. So it can the postsynaptic neuron, it can be any of those locations in the postsynaptic neuron. What's going to happen to the postsynaptic neuron in this example is that we have these neurotransmitters binding to these ligand-gated ion channels. So these ion channels are going to open in response to ligands. These are going to cause a graded potential because it's local and it may not cause an action potential, right? These aren't voltage-gated sodium channels, they're ligand-gated ion channels. So when these neurotransmitters, it looks like this is Oh, no, we actually don't know what they are. Once they bind these proteins, here we can see these two little red dots where they've bound that ligand-gated ion channel. Then the ions, whatever they are, either sodium or potassium or chlorine, what have you, flows into the cell. Looks like they're a positive ion. They're probably sodium ions because it says in this diagram that they're causing depolarization. And that depolarization, it looks like, is going to be strong enough to actually cause an action potential. So this depolarization is actually going to take us to our threshold of negative 55 millivolts. It could depolarize the cell. It could hyperpolarize the cell. And in that case, we're going to have either our EPSP or IPSP. That's where we get into these two, where if we have a depolarizing postsynaptic potential, we call that an EPSP because it's going to excite the neuron. It's going to make it closer to hitting an action potential. It doesn't always hit the action potential. So sometimes when we have a chemical synapse triggered, it will cause an action potential in the receiving cell. Sometimes when we trigger a chemical synapse, it's just going to make this neuron more excited. So it's going to be more likely to go into an action potential, but it's not guaranteed. The opposite of an EPSP, as we also talked about in lecture on Wednesday, is what we call an IPSP. It's just so many acronyms, just so much fun. An IPSP is an inhibitory postsynaptic potential where it's going to hyperpolarize the receiving cell. So that postsynaptic cell is going to get hyperpolarized, it's going to get pushed away from the threshold, and as it gets pushed away from the threshold, it's going to make it less likely to fire an action potential, which is going to be overall an inhibitory action, which is why we call it inhibitory postsynaptic potential inhibitory, because it's pushing it away from the threshold, and a excitatory postsynaptic potential is excitatory because it's pushing the cell towards an action potential. So an EPSP is pushing it towards negative 55 millivolts, and IPSP is pushing it away, and sometimes you have both of them active at the same time. One important follow-up from this wasn't in the question, but it's still worth addressing. An important follow-up is here. If you release the neurotransmitter just into this area physically, it will stay there. It will stay there unless something happens to it. And if you want to keep stimulating this postsynaptic neuron, that's a good thing. But if you want to just stimulate it once and then very specifically stimulate it later and not have it stimulate multiple times, you have to be cautious of where your neurotransmitter is going and how long it's staying there. So removal of neurotransmitters from a synaptic cleft is usually going to be very important. And usually there's a lot of space in here. This is their floating in cerebrospinal fluid. This neurotransmitter is just going to float away. So that's the first way that we're going to see neurotransmitters get removed from the synaptic cleft, is that they're just going to diffuse away. They're really highly concentrated here. They're not concentrated in other places, so they're just going to float away into other parts of the spine or the brain or the nerves. We can also have them be degraded, as we saw with the neuromuscular junction. This is not a neuromuscular junction, as we can see, because these are two neurons, but we're familiar with this idea with acetylcholinesterase, where we actually degrade the neurotransmitter. And so if we actually have a really high activity of our acetylcholinesterase, that's going to make it so that our signals will never get to where they're trying to go, which can be a good thing, just gives us more levels of control of if we're going to actually have action potentials fire from this postsynaptic neuron or not. And the last one that we talked about when we watched the Zoloft commercial is that you can reuptake the neurotransmitter into this presynaptic neuron, or the local glial cells can uptake the neuron, or they can just eat the entire neuron. The local glial cells can uptake the neurotransmitter. The glial cells are not nearly big enough to actually eat an entire neuron. So the glial cells are going to reuptake the neurotransmitter. They might give it back to the presynaptic neuron. They might degrade it, do something else with it, but it just gets removed. Either way, the big goal is that it's getting removed from the synaptic cleft. So if you have situations where in cases of depression, you don't have enough serotonin going through in your synapses, instead of asking your brain to produce more serotonin or more dopamine, what you're going to do is you're actually going to just inhibit the reuptake ability, so that you're producing the same amount, but the message is actually getting there. It's not getting shushed and pulled back into the cell before it actually makes it over to fire that active potential. So Prozac and Zoloft actually work in very similar ways in that they are reuptake inhibitors. So it's not that you're making more neurotransmitters, it's not that you're necessarily firing more presynaptic action potentials, is that the message is actually getting across the synaptic cleft before these neurotransmitters are getting pushed back into this presynaptic neuron. Okie doke. Okay, up and down regulation. Already did that. Never mind. Let's do. I've got another question in here. Yeah, if any of you have questions, there should be a live chat that you should be able to see. I'm hoping that all of you are able to see it. If it's not visible, I guess let me know by emailing me. But it looks like Kaylee can see it, so that's good. I'm hoping that it's working properly. My streaming software looks like it's working fine now. It's just a matter of if YouTube is working fine. I feel like one of these can't go by without at least something going wrong. I shouldn't have said that. Now I've jinxed myself. Anyway, I'm going to switch over to doing the blood-brain barrier. So let me pull that one up. Blood-brain barrier. And how do we maintain the blood-brain barrier? Well, I'll go over how it's formed, and then I can go over a bit of how it's maintained. Let me pull those slides up. All right. So when we're talking about the blood-brain barrier, we understand that our blood-brain barrier is important so that you don't die, so that you don't get exposed to nasty toxins and waste and other things that are not supposed to make it into your brain. So as we know, big things like proteins, a lot of waste, some hormones, antibiotics can't pass, but amino acids and sugar and electrolytes, those really nice ions, alcohol and anesthetics can cross over. So as far as how the blood-brain barrier is maintained, that's kind of a additional question. First, I'll mention how it's formed, and that information is found right down here. So it's formed by these really tight junctions that seal together the epithelial cells of the capillaries. So capillaries, we haven't done the cardiovascular system yet, but capillaries are where basically your vasculature gets pretty thin, and there are epithelial cells, also called endothelial cells, because they're lining the vasculature. And they get pretty thin, and in some places there are actually physically gaps between the cells so that big things can get from your bloodstream out or from other parts of your body into your bloodstream. But here, those epithelial cells that actually form the capillary wall are very tightly sealed together, and the basement membrane that is the basement membrane. Again, we haven't really talked about this, especially not recently. There is a basement membrane for the epithelial cells, and in some places it is broken. In the brain, it is continuous. So we have a continuous basement membrane that is an additional layer of protection. So we have these continuous sealed together epithelial cells, and then we have this basement membrane on the bottom of them that's sealing everything in all the way through. And then one of the most important things that we'll talk about in a second in detail is that we have the astrocyte cells, those microglial cells, covering the capillaries. So we have the processes of the astrocytes actually helping us to maintain the blood-brain barrier. Because just this alone, just the capillaries having those cells close together and having a continuous basement membrane, that's not enough to actually form our blood-brain barrier. So we can actually go over to a different set of slides here and see that our one of our glial cells, this is in our central nervous system, these are our astrocytes, these are going to form the blood-brain barrier. Here's our astrocyte here. We call it an astrocyte because it looks like a star. Astro meaning star, cyte meaning cell, so it's a star cell. Congratulations, astrocyte, you're a star. That was a terrible joke. The processes of an astrocyte are actually going to stick on to the blood vessel here. So they're going to cover the capillaries, and they're going to help by secreting chemicals that maintain the selectively permeable nature of the cells of the capillary. So they're going to actually, the maintenance of the blood-brain barrier falls on the astrocytes. Their job is to maintain the blood-brain barrier. So what we just talked about is how the blood-brain barrier is formed, but the original question was, how is the blood-brain barrier maintained? And that answer to that question is this slide. Astrocytes maintain the blood-brain barrier. They're the ones that actually help to regulate the chemical environment and ensure that nothing in here that's not supposed to get through is getting through. So they're very much the gatekeepers from the blood into the brain. And then the things that are actually producing the CSF, as we know, are a different kind of glial cells. Those are ependymal cells. These are the ones that are actually going to be making the cerebrospinal fluid that's getting circulated. Again, most of them are ependymal cells. They have the cilia, so they can beat the CSF around in the brain and in the spinal cord. But again, take-home message, maintenance of the blood-brain barrier, that's going to be the job of the astrocytes. They're going to be the ones who are maintaining that barrier and keeping the things in the blood that are supposed to be in the blood, and the things that are supposed to be in the brain, in the brain. All right, give me 1 second. Just poking around to make sure. Oh, yes, my live chat is visible. Okay, good. Okay, okay. Next question. Presynaptic facilitation and inhibition, and what neurobiochemical is involved? Let's pull that up. See, presynaptic facilitation and inhibition, that's also going to be with action potentials, I think. Aha. Okay. With presynaptic facilitation and inhibition. So we talked about, I'm going to just back up for 1 second to get to presynaptic facilitation and inhibition. We got to talk about GPSP really quick. So when we were talking about EPSP, our excitatory postsynaptic potential and our inhibitory postsynaptic potential. So if we have our postsynaptic cell hitting a small EPSP, we might reach, negative 56 millivolts, but we're not going to actually hit our threshold. We could potentially hit our threshold and generate an action potential, or we can make it more negative and actually go away from hitting an action potential. So the cumulative effect on what is happening is called the grand postsynaptic potential, because the neuron can be getting a lot of signals from a lot of cells at once. And so here's a picture of kind of what that looks like. So here's our postsynaptic neuron with all of these presynaptic axon terminals just sitting on it, where there's a lot of information you can be getting at once. So maybe you're getting, 12 different inhibitory postsynaptic potentials and 13 different excitatory postsynaptic potentials, and maybe that causes your overall millivoltage to go up from negative 70 to negative 60. That increase is going to be your grand postsynaptic potential. But there were a lot of things contributing to that. So what actually determines your grand postsynaptic potential is going to depend on your presynaptic facilitation or inhibition. And so there can be a third neuron forming a synapse with the presynaptic neuron at what we call the non-synaptic site. So that third neuron is going to release neuromodulators, which I don't believe we didn't talk about. We didn't talk about what biochemical that is. We just called them neuromodulators and left it at that. And they're gonna change the actual amount of neurotransmitter that's released by a presynaptic neuron. So this isn't saying that the action potential doesn't happen. This isn't saying that we're going to, make more action potentials. This is actually going to be messing with the amount of neurotransmitter that's being released from that presynaptic neuron. So here's what that's going to actually look like, where we have, here's our postsynaptic cell right here. And so what we see, this example, is we can have this neuron over here, B, that is going to be inhibiting our presynaptic neuron A. And so B is actually, it's not even touching this postsynaptic cell, but it's indirectly going to interact with this postsynaptic cell, because this cell A, is going to have an excitatory effect, as that plus sign. So if A fires, it's going to have an excitatory effect on this neuron here, neuron C, that's our postsynaptic neuron. And we have this other neuron here, this is neuron B. So we see when A fires, we do have a slight depolarization, and it's a graded potential, so we're going to get a depolarization, and then it's going to stop. And then if B is active, what B is going to do is it's going to inhibit, or it's going to, that is the right term, it's going to inhibit this neuron here. So this is going to be a presynaptic inhibition where it's going to decrease the amount of neurotransmitter released by this neuron, which results in we get no change in resting potential. So this one is actually going to be inhibiting this neuron right here. And for presynaptic facilitation and inhibition, it can be either way. We can either get facilitation like we just saw, or sorry, inhibition like we just saw, where it's going to decrease the amount of neurotransmitter that we release, or we can get facilitation where it's going to increase the amount of neurotransmitter release. And again, this is separate from how many active potentials it's going to fire. This is going to just be the amount of neurotransmitter that's released, which can also be modified by how many active potentials you're firing. If you fire more active potentials, you would potentially release more neurotransmitter. But this is a separate kind of control valve on the overall control that we have in the nervous system of exactly what is that grand synaptic potential going to be. So here's our nice summary slide for our grand postsynaptic potential, which is that in any given synapse, our neurotransmitters are going to have one of two functions. They're either going to be excitatory or they're going to be inhibitory. And here are neuromodulators. Those are going to be interacting with our presynaptic neurons. They can alter the rate of neurotransmitter release. They can also alter the amount of neurotransmitter release. And then this transmission can also be facilitated or inhibited by other chemicals as well. We didn't get into this a whole lot. But again, big takeaway, there's a lot of control, a lot of levels of control, a lot of levels of complexity in this system. And the effect of the presynaptic neurons can be altered by other neurons, including our presynaptic facilitating or our presynaptic inhibiting neurons. And any of those is going to modify our grand postsynaptic potential. So we can modify our grand postsynaptic potential by either firing an excitatory neuron or firing an inhibitory neuron, which is not shown in this diagram. Or we can have a presynaptic inhibition that then inhibits the excitatory neuron. So maybe due to two signals, we are wanting to get this neuron C up to its threshold potential. And to do that, what it looks like we would have to do is we would have to fire D and A at the same time. Because if we fire just A or just D on its own, we don't actually hit that threshold potential. But another part of the nervous system knows that we should inhibit that. Instead of inhibiting the postsynaptic cell directly, this neuron or this axon terminal B can just inhibit this one A. So even if D fires and A is supposed to be firing, it's not releasing enough neurotransmitter or it's not releasing it fast enough to actually trigger an action potential. We can imagine there are also going to be potentially inhibitory axon or inhibitory neurons touching this postsynaptic cell, and they could be being fired as well. And again, all of that is going to affect the grand postsynaptic potential of the neuron. Okay, and that's presynaptic facilitation and inhibition. Let's see how we're doing. Oh, my face is down so low. Let's see how we're doing. We're doing good. All right. Looks like we've got a couple people here. And I am out of questions. So if you would like to email me a question or put one in the chat, that would be much appreciated. Without questions, I have nothing to go on. So let's see. Looks like there's nothing in my inbox for my email. So if you have a question, go ahead and get it to me as quick as you can. Otherwise, I won't have anything to talk about, and I'll just sit here staring at the internet. Alright, looks like no one is around anymore. Maybe one of you, and maybe one of those one of yous is me, probably. So I'll give it about another 5 minutes or so. If I don't get any questions in the next 5 minutes, I'm going to go offline. If I get any others before 8 o'clock, I'll stream a little bit more and answer the questions. But if not, I wish you all the best of luck studying. If you don't get your questions in to me in time to have them streamed, I will probably reply by email. I'll be checking my email until pretty late tonight. So if you have any questions about the content or the material, you can always email me and let me know, and I'll get back to you. But other than that, I'll hang out for about 5 more minutes. Ooh, hydrophobic versus hydrophilic? Yeah, I can do that one pretty quick. So when we talk about things that are hydrophobic and things that are hydrophilic, what we're saying is, do they like water or not? And the reason that things like water or don't like water is because water is polar. So water, the oxygen and the hydrogens aren't very good at sharing electrons. The oxygen is very greedy. It will pull the electrons away from the hydrogens. So we get this partial negative side and we get this partial positive side. So if things are partially charged or fully charged, water is really going to like them because that negative part of the oxygen is going to be attracted to a positive, and the positive part of the hydrogen is going to be attracted to a negative. So anything else that has oxygens in it, like glucose, or any ions like sodium and potassium, are also going to like water. Those are all going to be hydrophilic or water-loving because they have a charge or a partial charge. Versus things that are hydrophobic, we're mostly going to see things like fats and lipids because they're just a huge sequence of carbons and hydrogens and carbons and hydrogens. And carbon and hydrogen are really good at sharing their electrons equally, which means they don't have any areas of charge, they don't have any areas of partial charge. So when they interact with water, water is going to be polar and have a charge. Lipids and fats are not going to have a charge, so they are not going to get along, which is why when we see water and oil in the same container, they will separate out because the fats do not like water. They are hydrophobic. So anything that is not polar is hydrophobic. Anything that is polar or charged is hydrophilic. Alexis, to answer your question, yes, I did get Julia's email and I answered all of the questions. You can go back in the stream, or once the stream is over, go back to an earlier part of it, and all of the questions are answered. All right, looks like there's no other questions for now. So I'm going to go ahead and log off. If I get any more in the next 30 minutes or so, I'll go ahead and stream a little bit more so I can make sure I get all of you your answers. But again, if you don't catch me in the streaming time and you would like to still get some answers, go ahead and email me and I'll probably email you back within a couple hours. Best of luck to all of you studying, and I hope you have a wonderful night. I'll see you tomorrow.